Okay, the neuron. A multipolar neuron. So a multipolar neuron is a type of neuron which is frequently seen in the central nervous system. Here we have a picture of it. So we have the soma, the cell body. We have the dendrites. These are the dendrites right here. Right? Okay? And etc. all around the cell body. And then you have the axon right here, which goes all the way to the end. You have the axon terminals, which are these little things right here, the end of the axon. Because you have this branching that happens at the end of the axon. The axon sort of starts to branch into different paths and etc. And these are the axon terminals on the end. Okay? And of course the myelin sheet, which covers the axon of central nervous system neurons. The signal message is received by the dendrites and the cell body, the soma, then proceeds down the axons toward the axon terminals, which are attached to other neurons or effector cells. So this is what is being shown here, the direction of the message and how it travels. It always travels in this direction. It comes into the dendrites and the cell body, goes down the axon, and it goes to the axon terminals. Okay? Note, signals received by dendrites will travel to the soma and then proceed to the axon. Axon length can be 1 meter long, and dendrite length is sometimes much larger than the diameter of the soma. We will be discussing multipolar neuron exclusively, but the mechanisms that govern its actions are shared by other neuron types. So once you understand how this neuron works, then when you look at the other neurons, it's sort of like, oh, the neuron is just put together a little bit differently, but the same things apply. So just to be clear, and let me get rid of this stuff right here. So the soma is this part right here. Everything in the cell, in the neuron that is not the axon, but minus the dendrites. And the dendrites would be, of course, these things right here. Right? So messages will be coming in from other neurons into the dendrites and the cell body, the soma, and then that message is computed, and then it is shot down the axon toward the axon terminal branches. Okay. In our quest to learn how a neuron works, we will start with the axon, move to the axon terminal branches and the neuronal connections to other cells, and then finally, how the signal received by other neurons is processed by the soma and dendrites and passed on to the axon. So basically, if we go back to this picture that we had above here, so first we're going to deal with this part right here. The axon, right, and everything here, here, and how the signal travels down the axon and eventually gets to the axon terminal branches. And then the second part, we're going to deal with what's happening at these axon terminal branches and how they're attached to other cells and what that attachment is like, and etc. And then we come back, because these attachments, if it's attached to these axon terminal branches, if they're attached to another neuron, then we're going to deal with, well, what is the attachment doing here? Right? And how are those signals coming in to the cell body, the soma, and the dendrites? How is that calculated into sending a proper signal down the axon, the signal that we're going to be talking about first? Action potentials. The signal which travels down the axon is electrical. This electrical signal is created by changing the potential on a patch of membrane from a resting potential of minus 70 millivolts to a positive voltage near or at sodium's equilibrium potential of about 60 millivolts. This change in membrane potential from resting potential to sodium's equilibrium potential travels down the axon at a speed of 100 meters per second. As the signal moves down the axon, the membrane behind it is repolarized back to the resting potential. So before moving on, let's explain what these words right here, repolarization and depolarization, mean. So if I have a membrane here and I'm at resting potential, now I'm not going to put in the charges like I usually did, because if you have minus 70, you know the inside is more negative than the outside. And this will be my extra here and cytosol here. And let me just put this here too. So minus 70, we're at resting potential, right? And if the membrane goes from minus 70 to near sodium's equilibrium potential, let's say 60 millivolts, this is called depolarization. Now, the process of going from 60 millivolts back to resting potential is called repolarization. Depolarization, repolarization. Okay, and there's another variant of this that we'll be discussing when we get to it, but not right now. Okay, so a patch of membrane is depolarized from resting to sodium's equilibrium potential, then repolarized from sodium's equilibrium potential to resting potential, but the adjacent patch of the membrane on the axon terminal side is then depolarized. This cycle repeats as the depolarization moves down the axon's membrane. This is a description of an action potential. So this is our first description, okay? And we're gonna, of course, get deeper into how this happens. So let's look at what we mean by this. Okay, so we have our axon, and here's my cell body, right? And it's got its dendrites and etc. going off. And, right, and I'm trying to do my best to draw a good picture for you guys, but, you know, it's not easy. And here's my axon going off, right? And it's eventually going to go to the terminal branches and etc. We're not really worried about that right now. So an action potential will be generated right here at the beginning of the axon and then travel down all the way to the axon terminals. Action potential, we'll just put AP. Now, what is happening when I just look at a patch of axon membrane as an action potential is traveling down, for example, let's say this patch right here. I really go in on it. What happens is that this patch, if I'm just monitoring it as the action potentials are traveling, this patch will go from resting potential of minus 70 to, let's say, 60 millivolts, 50, 60 millivolts, near sodium's equilibrium potential, and then go back to minus 70. And then it will go back to 60 millivolts again. And then you just kind of go around and keep repeating. So when I'm just looking at that one patch, if I have action potentials traveling down the axons being generated right here, right, then the patch will simply change its potential, that small patch, from minus 70 to 60 to minus 70 to 60. So depolarize, repolarize, depolarize, repolarize. Now, if I look at the patch right next to it, so let me go ahead and erase this arrow right here. So if I look at the patch right next to it, then what I'll see is if I'm looking at what's happening on this patch, then this patch will actually be doing the same thing, but it'll be 60 millivolts, then minus 70 millivolts, then 60 millivolts, then minus 70 millivolts. Meaning that the potential on that membrane is going to be the opposite of the potential to the membrane next to it. So this membrane right here, which I should have maybe wrote the numbers in red, but let me just go ahead and color it black right here so we know that's what we're talking about. This one right here, you can see when this membrane is repolarized back to resting, the neighboring membrane next to it right here is depolarized. And then when this is depolarized right here, then the patch to the right, to the axon terminal side, is actually repolarized. So it's ready to be depolarized again. So you have to sort of look at this. This is your initial gateway into what an action potential is doing. And that is that when you're looking at two patches of membrane right next to each other and an action potentials are traveling down it, the patch prior on the somatic side, meaning over here, right? When this is set to minus 70, the patch next to it is going to be depolarized. And then when this patch next to it, the green patch, is repolarized, the patch on the somatic side, the somatic side is going to be depolarized. And this sort of switching continues. And why is it important that the patch that we have right here, why is it important that if the patch prior on the somatic side is depolarized, that the patch on the axonic side, right next to it, axonic terminal side, right next to it, is repolarized? Because once it's repolarized, the green patch, then it can be depolarized again. Okay? So it's a lot to unpack. To start thinking about what's happening, this is where you start. The generation of an action potential is due to the actions of two voltage-gated ion channels, the voltage-gated sodium channels and the voltage-gated potassium channels. Both channels have very similar gating mechanisms. Both channels have three different states: open, the conductor ion when they're open, inactivated, not conducting their ion, closed, not conducting their ion. Both channels cycle through these three states in that order, as in open, inactivated, closed, open, inactivated, closed. So it's never going to be open, closed, and then inactivated. It will always be open, inactivated, closed. Then you go from closed to open. Then once you open, you become inactivated, and then once inactivated, it can go to the closed state so that it can open up again. Both channels will go to the open state when the potential on the membrane they're situated in moves from resting potential towards sodium's equilibrium potential. So as the membrane starts to depolarize, remember, depolarization is just going more positive than the resting potential. It doesn't necessarily mean that you're already at sodium's equilibrium potential. As you go from minus 70 and you start moving up towards zero, that is depolarization. So as that patch of membrane starts to depolarize, those voltage-gated sodium and potassium channels can go from the closed state right here to the open state. Make that transition. Increased depolarization will increase the likelihood of these channels going from the closed state to the open state. Remember what we talked about with voltage-gated channels, ion channels, gating mechanisms in the previous part of this section? We said that if I had voltage-gated sodium channels, for example, as the voltage on the membrane starts to change more of those channels, the probability of these channels going from the closed state to the open state increases. So more channels will be in the open state, make that transition. So I can draw it out here again. We had conductance of sodium. So we'll just call it picoamps, because conductance of charge is just going to be amperage, right? And this is the potential on the membrane. So as the potential, we'll start out with minus 70 millivolts, and this is going to be in millivolts, and go all the way to plus 60. So we're going to go with a voltage-gated sodium channel. We're not going to worry about the inactivated state in this example. But this is the picture that I had in the previous section, that as you move up, what will happen is, as the membrane potential increases, the amount of sodium being conducted increases, right? And all that is saying is that eventually you'll reach some maximum level because there's only so many channels in the membrane. But what this is telling you is that as the potential starts to shift toward a positive number, more of these channels will go make the transition from closed to open. So a short period of time after being in the open state, the channels will become inactivated and no longer conduct their ions. And this is the part of this graph that we haven't drawn out, and we're going to draw it out in more detail soon, okay? So actually, let's erase this. So this transition from open to inactivated is random. Sometimes the transition is very quick. Sometimes the transition might be a little bit slower. But once the channels go from closed to open, they will automatically transition to inactivated state. And remembering that in the inactivated state, the channels don't conduct any ions. Inactivated is not closed. Inactivated just means that it's not conducting this ion, and it cannot go to the open state from inactivated. You need to go to the closed state. These are actually two different conformations of the protein, inactivated and closed. They're not the same conformation. That's why they're different from each other. Repolarization of the membrane will lead to these channels transitioning from the inactivated state to the closed state. So once the membrane starts to get repolarized, meaning it's moving back to the normal minus 70 millivolts, then these channels will start to transition from the inactive state right here to the closed state. And once it goes to the closed state, then it is ready to again go to the open state if necessary, meaning that you start having depolarization of the membrane, and then you start going back to the open state. The channel cannot open until it has transitioned from inactive to closed. Voltage-gated potassium channels transition from closed to open is slightly delayed. Thus, voltage-gated sodium channels will on average go to the open state first, and after a brief lapse of time, the voltage-gated potassium channels will go to this state. And this is important for what we're going to be talking about next. I mean, everything is important, but yeah, just keep that in mind. The figure below depicts how a voltage-gated sodium channel functions. So here we can see we're enclosed, right? And you can see you're positive on the outside, negative on the inside. The plasma membrane is at rest. And then you go to the open state, membrane is depolarized. It's kind of showing it graphically here. You're a little bit negative outside, you're positive inside, meaning that the membrane potential is starting to become positive. And it goes to the open state, right? And this is the open state right here. And then it starts to move to this inactivated state, right? The potential in the membrane hasn't necessarily changed because you're still negative outside and positive inside. But you can see it's sort of in that open state, but then you have this sort of arm coming in and blocking the channel. Now, to go from inactivated to closed right here, the membrane has to be repolarized back to the membrane's resting potential. Once that is done, then it can go to the closed state. Otherwise, if repolarization doesn't occur, it stays in the inactivated state. A voltage-gated potassium channel also cycles through these three states in a similar manner, just slightly delayed as compared to a voltage-gated sodium channel. So the voltage-gated sodium channel and the potassium channels follow the same pattern. It's just that the voltage-gated potassium channel is one step behind the voltage-gated sodium channel in going through this three-step process. All right, so we're going to stop here and then go over the action potential in more detail later. Okay, guys, thank you.